Intermolecular Forces, Part 2, Section C. Now we're going to examine unusual, or sometimes ignored, intermolecular forces that are very important in biological systems. First, we have the Pi-Pi interaction, which is a London force between the surfaces of nonpolar pi bonds, like carbon-carbon double bonds. It's most important if you're talking about a benzene ring, so that's where we're going to focus. Pi-Pi interactions are also directional. Our strongest Pi-Pi interaction is the Pi-Pi stacking interaction, where the benzene rings are parallel to each other, and that gives you the maximum overlap. Then we have the Pi-Pi-T interaction, in which the benzene rings are perpendicular or face to edge. This is a slightly weaker form of Pi-Pi interaction. Again, it's a London force. Polar pi bonds that are part of rings that have benzene-like resonance can also have Pi-Pi interactions, as we'll see in a moment. Here is a rotating structure of the double helix of DNA, the blueprint for all life on Earth. We're going to look at the types of attractive forces that allow these two strands to wrap around each other and form the double helix. Although the backbone of the strands in double-stranded DNA are made of covalent bonds, what causes the two strands to associate are non-covalent bonds. We have the 3 hydrogen bonds between the DNA nucleotide bases G and C, and the 2 hydrogen bonds between the DNA nucleotide bases A and T. These are specific interactions. G will always match up with C, A will always match up with T in double-stranded DNA. Now, between the rungs of the ladder, stacking one on top of the other, you see that G will have an interaction in red with A. That's a Pi-Pi interaction because G and A represent aromatic rings. They have benzene-like resonance. If you actually draw a resonance structure that shows the lone pair on nitrogen in G resonating up through the oxygen in C double bond O, you can see the benzene-like resonance. The same thing occurs between C and T. They stack on top of each other and have a Pi-Pi interaction, parallel Pi-Pi interaction, or Pi-Pi stacking. And again, if you take the nitrogen in C and resonate that through C double bond O, you will get an aromatic or benzene-like resonance. In T, you can do the same thing, but now you have to do it twice. The lone pair on the nitrogen next to C double bond O has to resonate to form a double bond, and the other nitrogen has to do the same thing to the carbonyl at the bottom left, and that will form another aromatic benzene-like resonance. So that would explain why you have Pi-Pi interactions between C and T. Being aromatic makes a molecule so stable that charge separation, even double charge separation as we see in T, doesn't matter. It still wants to be aromatic. This is part of the interior of HIV-1 integrase, an enzyme which is produced by the virus HIV-1, which causes AIDS. If you look at the structure of this interior portion, there are T-type Pi-Pi interactions between each of these amino acid residues, and these T interactions stabilize HIV-1 integrase. It's possible that disrupting these T-type Pi-Pi interactions could lead to the failure of the enzyme to function and thus act as a treatment for HIV infections. Cation pi interactions are impressively strong. They're up to 10 times stronger than a typical hydrogen bond, which makes them about half the strength of a normal covalent bond, and they're also up to 100 times stronger than the Pi-Pi interactions that we just studied. There are hundreds of examples of cation pi interactions in biology, and here's one. The neurotransmitter acetylcholine, which is responsible for nerve conduction in the brain and in muscles, is able to bind to its receptor through a cation pi interaction, which you can see between the tryptophan residue, part of the receptor's protein structure, and the positively charged nitrogen in acetylcholine. The formation of a positive charge in the molecule can also induce a folding behavior caused by intramolecular cation pi interaction, as we'll see in this example. Once we put a methyl group on the nitrogen and give it a positive charge, the benzene ring, which wasn't that interested in interacting through pi-pi interaction, now folds tightly onto the pyridinium ion, which is another aromatic ring, through cation pi interaction. The pi-pi interaction was not strong enough to actually cause the fold. Now we've come to the last of our unusual intermolecular forces, and this is the most ignored of all of them, and it's called halogen bonding. It's really too bad that people don't pay attention to this more, because halogen bonding is extremely important in understanding the interaction of many organic compounds with biological structures, as well as in drug design. So let's see how halogen bonding was first discovered. If you take the two liquids, 1,4-dioxane and bromine, and mix them together, they will form a crystalline solid. And you note in the picture, the bromine atoms are attracted to the oxygen atoms, and that links all of these strands together in the crystal structure. This was discovered back in 1954 by Dr. Ad Hassel, and he received the Nobel Prize in 1969 for his investigations of halogen bonding. We have a general definition for the halogen bond, which is an attractive force between the halogen bond acceptor atom X, which is nitrogen or oxygen with a lone pair, and the halogen bond donor atom Y, which is chlorine, bromine, or iodine, and it can't be fluorine unless it's F2, and we'll see why when we explain how this attractive force works. The lone pair on X is able to drill into the electrical field of around Y, pushing away the electron density such that there is a sigma hole created. That is, a region of partial positive charge near the nucleus surrounded by a crown of partial negative charge. It's this sigma hole that allows you to get a halogen bond, that allows an attraction, which is basically electrostatic, between the lone pair on X and the nucleus of Y. Fluorine is so electronegative that the lone pair on nitrogen or oxygen can't disperse its electron density and drill into its electrical field to make a sigma hole unless the fluorine is only bound to another fluorine atom. CK2 is a protein found in high levels in all human cancer cells, and if it's not functioning, the cancer cells simply die. They commit what's called programmed cell death or apoptosis. So it seems like it's a good idea to try to inhibit this enzyme, because if you can inhibit it, then the cancer cells should die, and that might be a great way to treat cancer. So if you look at our picture of CK2, there's a little molecule on the interior of this very large structure which has ball and stick representation, and that's the inhibitor. It's bound inside the enzyme, and it's going to gum up the works, preventing it from doing its job, and thus allowing the cancer cells to die. The red atoms in the ball and stick model are actually bromine atoms, and they're interacting with the backbone of CK2. Now our inhibitor is known as K25, so it's the first in a lead set of compounds for development of inhibitors of CK2 that could be used as potential anti-cancer drugs. Practice problem 3. Okay, the picture on the left looks pretty complicated. It has to do with an inhibitor binding to an enzyme. In the model shown, the inhibitor, GLC-CLU, is a thick stick model. Okay, so you see those thick bonds for the middle structure, that's the inhibitor. That's bound to the enzyme glycogen phosphorylase B, which would be a huge molecule, so we're only showing the portions that are very close to the inhibitor. And we're showing those as ball and stick models. The gray atoms are carbon, the blue atoms are nitrogen, the red atoms are oxygen, and the green atoms are chlorine. You want to assume that the nitrogens on the enzyme chains, those are the ball and stick chains, have NH bonds. So there's a hydrogen attached to nitrogen. We just don't show the H's attached to N. And that the 5-sided ring, which is at the bottom right corner, with 2 nitrogens, 2 blue nitrogens, that's imidazole, that has a positive charge. And that 2 O's, remember O's are red, attached to a C, which is gray, has a negative charge. It's called carboxylate. It's CO2-. And you can see that in the bottom left side, there are 2 red oxygens. One of them is kind of tilted back, attached to a gray C. The dashed lines are attractive forces between the inhibitor and the enzyme. You want to identify each attractive force. So what kind of attractive force is it? Is it a hydrogen bond, halogen bond, cation pi, pi? So that's the kind of thing that you want to do. So carefully examine each interaction based on this information to figure out which attractive forces we have. So at this point you want to pause the video and take a good look at the model on the left side.